Hello, welcome to Anatomy and Physiology. I don't know about you, but I'm definitely missing the classroom more and more. So I miss teaching in front of you guys rather in front of a computer. It's not the same, but it is what it is. Okay. Today we are going to be talking about physiology, a little bit of the urinary system. Okay, and we're going to get looking into the structure, the nephron. Now, our last quiz was on the anatomy, so therefore we're going to get into the structure here, the nephron, and this is really the structure that does most of it. Okay, the other structures really are just tubes in order to get what this all creates out of your body. We're going to get into it, going over it. You have the Bowman's capsule, this bowl-like glomerulus, capillaries inside. Okay, then through your PCT or proximal convoluted tubule, down through your descending limb here, up through your ascending with an A. Okay, those make up your loop of Henle, through your distal convoluted tubule or DCT and into your collecting duct. So we're going to go through all of those in this section, as well as urine formation. Okay, pretty much what's going to happen here is we're going to learn about how urine is made. Okay, we talked about why we need urine last section in digestion with ammonia. Okay, so we want to get rid of ammonia. How are we going to do that? Well, this is going to be the process in which we do. So the functions of the nephron: reabsorption of filtrate, reabsorption of water, and secreting any waste into tubular fluid. That waste in particular being of ammonia or urea. Okay, so fluid movement. First, it's going to start off as filtrate, then get into tubular fluid, and then finally into urine. So what's the difference between all of these? Well, that's what we're going to talk about. The anatomy, the renal corpuscle, that's going to be your capillary network and also your glomerular capsule. Okay, so that's going to be pretty much the blood entering in. Okay, then you have your renal tubule, your proximal convoluted tubule, nephron loop, which is being the loop of Henle, distal convoluted tubule, and collecting duct. So those are the structures. Let's get into each of them. The renal corpuscle, okay, this here is what we're talking about. You're going to have Bowman's capsule that's going to be around here. You're going to have your glomerulus here, or your capillaries that are going to be in here. And what? All right. So the capillary networks are going to be fenestrated. Okay. What that means is it's pretty much going to force the liquid through these microscopic kind of holes or filter system in here and collect it. Okay. So to enter the corpuscle, pores of the endothelial cells lining. So therefore we know they're endothelials. All right, fibers of the basement membrane and filtration slits. All right, so pretty much filtration happens because of the pressure difference. That's pressure is going to build up, okay, and pressure flows from high to low. So high concentration inside here, low concentration out. It's going to force that filtrate out of and into the Bowman's capsule here, and it's going to collect that. Then what's going to happen after that is going to be what is taken in and out. So after this, right, you're going to go into your next structure. That next structure is called your proximal convoluted tubule or your proximal tubule. Okay, it should not be loop here. It should be tubule. So that is a mistake. I will fix that. But it should be PCT, proximal convoluted tubule. All right, in here you're going to have the absorption of organic nutrients, plasma proteins, ions from the tubular fluid. Okay, so you're going to take back because right now you're forcing everything out and then you're taking back what you need. So you need organic nutrients, plasma proteins, ions from there. Okay, so it releases into the interstitial fluid. Okay, interstitial fluid is fluid in between cells. Okay, therefore the interstitial fluid is going to increase and the tubular fluid is going to decrease. So if I'll kind of draw a basic structure here. You have your cell, okay, you have your cell, and then you have fluid, well, in between. This is your ICF, okay, this is your ECF. Or simply put, you have interstitial fluid being you have fluid in between the cells, okay, and then you have fluid outside of the cells. Actually, this is going to be your ECF or your ICF, sorry. Inter meaning inside, ECF being outside, or this being tubular fluid. Okay, so I had mixed this up. Okay, so this is your interstitial fluid, okay, or intercellular fluid, same thing. Okay, and what's going to happen is the interstitial fluid is going to increase because it's going to move in here. Therefore, this is going to decrease. Okay, so your tubular fluid or your ECF is going to decrease, or the pressure of that's going to decrease because it's going to move into it, which therefore is going to increase the pressure there. So interstitial fluid is going to increase. Tubular fluid is now going to be decreasing as you're going to be uptaking the nutrients. Okay, you have the ascending limb. Okay, remember this is going to be, this is backwards here. So this is 1, 2 here. So from the distal convoluted tubule, you're going to have, you go straight into the descending limb. Okay, so the descending limb is permeable to water, and water moves out by osmosis. So once it is inside the descending limb, water is going to get pushed out through osmosis. Okay, and then water, you're not going to have a lot of water in your tubular fluid. Okay, so your ascending limb then is going to actively transport sodium and chloride ions out of the tubular fluid. Now that's key. It's actively transporting it out. You know that if you have a high concentration because the water's out, you have a high concentration of sodium or chloride or salt in general, that water is going to want to flow through. So what's going to have to happen is your ascending limb is going to have to push that sodium, push that salt out, so that way that water doesn't go back into the ascending limb. So you want to, when you remove the water, you want to hold on to the water. So you also have to get rid of the salt, and that's what happens here in the ascending limb through active transport. It's going to get rid of the salt, so that way the water doesn't go back in and you're not losing too much water. Okay, so to sum this up, descending, first get rid of the water, ascending, get rid of salt. Then you have your distal convoluted tubule, or your DCT. Okay, this is going to be active secretion of ions, acids, drugs, toxins, things like that. Selective absorption of sodiums, ions, and of sodium ions from tubular fluid. Osmosis happens to concentrate the tubular fluid. So what that's saying is that osmosis, okay, is going to happen, once again trying to take out the water. So this process is trying to concentrate the tubular fluid, as well as the selective absorption of salt. So your body is going to absorb more salt, try to take more salt into that, and then you're going to have secretion of ions, acids, drugs, toxins, things like that that are going to make up what is in your urine. Okay, so osmosis here, the importance of osmosis is to understand the concentration. Okay, so osmosis here is happening to balance it out. Okay, selective absorption of sodium, so that way your body's not losing that. We need sodium. And then active secretion of these things, right? These mostly are going to be toxins or waste that your body's not necessarily going to be using. And these could maybe have some to get rid of. Then you have the collecting duct. Okay, that is the final adjustments to urine composition. Will absorb water, need to, or absorb, secrete sodium, potassium, hydrogen, bicarbonate. All right, so this is pretty much mainly to finalize it. Okay, so if you're looking at this as a paper, okay, this is kind of your finishing touch. This is your finalized paper. So here's all the kind of breakdown of it. Glomerulus is going to be the filtration of blood, water, urea, glucose, amino acids, salt can pass, but but plasma proteins and cells cannot. Okay, so all of this is going to pretty much pass into the renal capsule. Okay, and it's going to collect the filtrate. So all of this, so in your filtrate, you have blood, water, urea, glucose, amino acids, salts. All of those are considered filtrate. So that gets collected through the renal capsule. It gets then moves through into the first convoluted or proximal convoluted tubule, where selective reabsorption of water, glucose, amino acids by active transport. So it's going through active transport here to maintain the levels there. Then you go through loop of Henle. You're going to reabsorption of water and salt. Second convoluted or distal convoluted tubule is going to be the secretion of waste into the filtrate. Okay, so your waste isn't getting secreted until the second convoluted tubule, mainly because you have to try to manage your water and salt. And then finally, your collecting duct, which is going to reabsorb water as it passes urine to the pelvis of the kidney, and then eventually out of the kidney. So going into kind of urine formation, those are the structures we need to know. That chart is huge. This has it kind of broken down a little bit more better understanding for you. You need to know this chart. Here's all the different parts of the nephron, and here's their functions, and this is pretty much how urine is formed. So a little bit more about urine formation, blood composition regulation. Okay, so excretion items, meaning what is excreted. Okay, urea, that is the toxin ammonia that gets converted into urea. That's that organic waste. So you excrete about 21 grams a day. Creatinine, which is a breakdown of creatine phosphate, about 1.8 grams per day. And uric acid is a breakdown and recycling of RNA, 0.48 grams. All of these here, okay, the reason for this, okay, all of these is from metabolism. So all of this process here is waste that happens because of your body trying to break down and make energy. So you create all of this, or you consume all of this, and now your body has taken it in. Your body goes through the cell processes and breaks it down. Now you have some waste. How is your body going to get rid of it? Well, it's going to get rid of organic or ammonia waste through urea. It's going to get rid of creatinine or the breakdown of creatine, and uric acid or from the recycling of RNA. Okay, so these are your averages here, but you can see the majority and the largest part being urea. You actually also secrete urea through your skin as well in sweat. Okay, so it has the ability to be secreted through your skin. It's very in small dosage, but you can secrete urea through your skin as well. Okay, so a little more into urine formation. First off, remember we talked about it first. Okay, filtration. You're going to have to get that filtrate. Okay, the pressure forces water across the filtration membrane. This occurs in the renal corpuscle. Okay, then you're going to have reabsorption. Okay, reabsorption is the removal of water and solute or salt and reentry into the circulatory system. Okay, this is going to happen primarily in the proximal convoluted tubule, primarily in the PCT. Okay, also the loop of Henle as well. That is going to there. So the loop of Henle as well, the descending loop taking in water, the ascending loop reabsorbing salt. Okay, so those two structures, and then the secretion happening in the um, DCT or distal convoluted tubule. So that's going to be the secretion of the waste. All of what you just saw last slide is going to be secreted in the distal convoluted tubule. So you may see questions as far as which structure or what structure is ammonia or urea going to be secreted in during urine formation. All right, so you have to understand a little bit as far as what and where that is going to happen. Now, here is a understanding of the different concentrations of plasma. Okay, this is meaning in your blood. Glomerular filtrate, this is that filtrate, and then finally your final product of urine. Okay, so urea, you can see that in your blood, not so much. Okay, 26 percent. So we're looking at moles per equal liter. So you have 26 here, glucose 100, glomerular stays the same, and then you can see the big change here. Okay, if you look at these wastes, a lot in the urine, and then these are nutrients, right? So plasma and are nutrients, but all of this gets, for the most part, pushed in or pressurized through that, and then you're going to get a deposit of that here out of your body. Okay, so you can see here as far as concentrations in urine versus filtrate and blood for all of these. Sodium, potassium, calcium, magnesium, chloride, bicarbonate, sulfate, phosphate. So these here are what are considered healthy levels here. So if you saw increases of this, then you can have an understanding of what your body is going through. So a increase in glucose in urine is a sign of diabetes. So that's something to have an understanding as far as what you can see with that. Okay, so another kind of your normal averages with urine. It does have a pH 4.5 to 8. Your average is about 6. Okay. Specific gravity. We can go into that. We were going to do a lab, but because we're at home, we're not able to do that. But it has to deal with kind of buoyancy, I guess, has pretty much the force of gravity on it. Then you have osmotic concentration, 855 and so forth. Water content, somewhere between 93 to 97% water. And just think, that's us absorbing water twice, right? Reabsorption of water happening, okay, and the PCT as well as the descending loop, and then other places as well. So depending on how much water and things you have there. So volume, as far as how much you urinate a day, anywhere from 700 milliliters to 2,000 milliliters, or to 2 liters, or 0.7 to 0.2 liters per day. Color should be clear yellow coloring. Odor. Now, there's genetic properties that can be this as well, usually through coffee and asparagus. Asparagus is a genetic, passed down through the genes. If you eat it, your urine tends to smell. Coffee is one that, as well, is if you drink it, your coffee, some people, it will smell as well, and should be sterile. Now, there are stuff and practices. You'll probably have a question on this sometime for one of your uh, questions of the day. It's called urine therapy, where people drink their own urine. So a lot of different things. That's more of an Eastern type medicine. It is growing popularity as far as um, people doing it. Now you have to consult the doctor and to do it properly. But there is such thing that it's called urine therapy. So it is sterile. Okay. So that is pretty much it. All right. So we will talk more so about hormones and homeostasis the next time. Pretty much next week, your chapter for that, or your quiz for that, is going to be homeostasis. So there'll be a lot into that. We'll be going over this chart here. Very, very, it looks very complicated. But for today, we learned about the uh, nephron, how urine's made, and specifically what is in urine. So thanks for tuning in and enjoy your week.